All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So the discussion today is fascinating and important. I am seeing that so many folks are actually concerned about this. They have been talking about the gene therapy that is there, or the unknown effects of the vaccine, or the long-term reprogramming of the vaccine, and so on. So my request to the cool beans here is that please, I need your attention today to go over this paper. There are a lots of dots to be connected. This is all actually good. I like the study here. It has nothing dangerous in it, but it is important for us to understand the mechanisms they are talking about and if we should be concerned about that or not. So just need your attention. And if at some point I do not explain well enough, please ask me to repeat it. I would do that. So let's start. So this is the preprint. It says the BNT162b2, which is the Pfizer's BioNTech vaccine, messenger RNA vaccine against SARS-CoV-2, reprograms both adaptive and innate immune responses. So that is the basic title here. And we're going to go over the whole paper. I have the paper open here, highlighted and drawn. So we'll talk about that. I want to show you some of the concerns. If you go down here and look at the people's comments, you would see how much concerned people are. And I had actually tweeted this paper out over this weekend. And somebody said that on one end, you are not convinced that the vaccines are gene therapies, something to that effect. And on the other end, you are sharing such papers. So I think it is important for us to understand what this paper is. Then, so that's a bad joke. Yes, still isn't he. And then you would see a lot of questions here. mRNA vaccines are actually reprogramming the immune system. My concern as a worst case scenario, speculative, is the immune system waits for artificial prompt, fails to act normally, and an AIDS-like scenario plays out. So there is just tons of folks who are concerned, who are worried, who are upset, and saying, see this one, still believe in vaccination, and so on. So I think it is important for us to look at this mechanism. Let's start. I'm going to start with my artwork. This is, I think, 3 or 4 weeks ago. I had tweeted this out as well. So this is the sea storm, or the storm at the sea. So this is the artwork. Thank you for for being able to see my artwork without much criticism. So here is some of the links. These are in the description as well. So this is peripheral blood mononuclear cells. What are these cells? Then TLR4, we'll use this discussion, this topic in there as well. Toll-like receptor 4 are receptors on the innate arm that can identify or detect gram-negative bacteria. So with TLR4, what I would love if you can keep in your mind, bacterial infection. Then TLR7 and 8, if you can keep in mind viral infections. Then there is a study here, Interferon Gamma at the crossroads. So we'll look at that as well. And then another study here, or a paper here, innate immune memory, time for adopting a correct terminology. And this is 19 April 2018. And there is a lot of things I've highlighted here as well. With this, let me just provide a little bit of pretext or context to this paper and the discussion. The context is actually very simple. The context is, we know for decades, centuries maybe, century at least, that there is a adaptive immune system, or humoral and cytotoxic immune system, which has long-term memory. And we have discussed that many times over here as well. That long-term memory is brought together. We're talking about adaptive arm. It is done, that memory is established by keeping the copies of the cells that have resisted or encountered some antigen and responded to it. So our body keeps copies of those cells that are fighting against someone antigen, and then it uses them in the future. One function of the vaccines or actual infection, one outcome of the actual infection and one function of the vaccine, is to train the adaptive immune system to produce those memory cells to defend against that antigen in the future. And not only just defend, but defend rapidly within 24 to 48 hours. So before the pathogen that comes into our body in the future, before it can cause too much damage, our adaptive arm gets up, wakes up, responds, and eliminates the pathogen, in theory, in ideal situation. Now here is the important part, so please focus on this one with me. For decades, or even for a century, there has been a lot of curiosity that how does the innate arm learn? We know, we have an idea of how adaptive arm learns. We know it creates memory cells, but how does the innate arm adapt itself and adjusts and learns? That has been a very important area of which is unknown, plus which is the target of a lot of studies and research. And there have been studies since 1940, not new, since 1940, talking about the innate arm learning, which we say reprogramming. We used to call it reprogramming, learning to adapt against the newer antigens and learning to fight with them without creating memory cells. And now here is an important part. It may be that innate arm does create memory cells. We are just not aware of those yet, just like we are aware of the adaptive arm. So we may actually, after 10 years, 15 years, 20 years, maybe we'll know more. But so far what we know is that the innate arm does not create memory cells, but instead it learns to respond to various antigens that it encounters throughout our life. That response can be either an enhanced response to some antigen, and that is called trained immunity. This is a term used for the memory. I'm using the word memory flexibly over here. This is a term used. I'm going to stop sharing and just bring myself to the screen for a second. This is a term used for innate arm, where we say it learned to respond to some stimulus when it encountered it with force, with more reaction than it did the first time. And if that happens, if the innate arm's response to an antigen increases in the future, then we say that that is a trained immunity or priming of the innate arm. If the innate arm cells learn to respond less to an antigen, then we say that innate immune tolerance has developed. The same mechanism on the adaptive arm is called anergy, A-N-E-R-G-Y, or immune tolerance, where our immune system ignores something and says, you know what, I don't care. So adaptive arm, I'm going to conclude this discussion and then go to the paper. Adaptive arm is known to have memory. Adaptive arm can respond with higher intensity or lower intensity. However, innate arm, we are not very much clear. We know this happens, but we do not know how. And so the mechanisms are named. We call it reprogramming of the cell where the genes are expressed, or we call it innate arm tolerance or innate immune tolerance, where the genes are not expressed too much, or cross-training or priming or training, immune training. These are the innate arm terms. So this is the basic idea. This idea is known since 1940. It's not a new thing. It's not that today we are discovering. Infections cause that. That is how in the future our innate arm responds better to some infections. Also remember, innate arm is non-specific. Non-specific means innate arm doesn't care for what is the epitope on a pathogen. Instead, it responds, it has the pattern recognition systems. It still works with the patterns, but not with the specific epitopes. So we call it non-specific response. Although we know innate arm can actually be specific too. And for various kind of specific patterns, it responds in various ways. We do not know how does it do that. And so there is a lot more to be learned about the innate arm. So with this background, the quick summary of this paper that we look at. The paper was the following. Authors said, now this is, please, once again, I need your focus. This is an important point. Author said, we know BCG vaccine or MMR vaccine, measles, mumps, rubella vaccines, they cause reprogramming of the innate arm. They train the innate arm to learn to respond to these pathogens that we are giving as vaccine, to respond to them in the future. And they say, the thing is this, we know about this, and we know that these vaccines, BCG and MMR, are inactivated viruses or inactivated pathogens. They said, now we have a new vaccine, messenger RNA vaccine. It is not inactivated pathogen. It is not a partial protein like Novavax. It is actually a new technology. So we were curious, the authors. They said, we were curious that we know it is creating memory cells in the adaptive arm. Is it helping learn on the innate arm side that innate arm should become primed for SARS-CoV-2 in the future as well? Is it helping innate arm learn as well? Is this vaccine doing that or not? That was the basic question in their mind. So please realize this, and I'm going to show all of this in the paper. So the concept of this is a gene therapy, and then we have a genetic change, and the big pharma is sending in some little nanobots, and that's going to go and change some genes, and now we are doomed forever. No, that's not the case. So let's now look at the paper and the drawings. And please forgive me if I look like I'm paying more attention to this. I know you can go through this paper, you can learn it by yourself as well. So it is not at all in any way supposed to be that I don't trust you can get it. It's just that when I want to do it, I want to make sure that we are on the same page. So this is the gift for somewhat nervous humanity today. And let's see what we have. We are saying that the Pfizer-BioNTech reprograms. That's the word used, reprograms. And this is a word used for decades. It's not a new word. Innate and adaptive arms of the immune system. So the question is, are we in trouble? And then have we proved now that the vaccines are gene therapies? To me, somebody even sent a message that, have we now proved to you that Geert was correct? And actually, very far from those proofs. So the question is, are we really now dead? Have we proven it that the vaccines are now just going to kill us? So let's see if that is the case or not. So summary is the following for this paper. Summary is that the vaccine, Pfizer-BioNTech vaccine, effectively causes humoral and cellular immunity to become effective, or to have its response. So what are they saying? Here is, I think the cool beans who've been with me for now about a year, you know that there are two parts of the immune system. I have just represented them over here, not all the cells and proteins are here. So this is the innate arm, non-specific arm, and this is the adaptive arm. In the innate arm, we have natural killer cells, we have dendritic cell, we have macrophages, we have neutrophils and so on, monocytes and so on. In the adaptive arm, we know we have helper cells, which are T helper cells, and then we have cytotoxic T cells, and we have B cells. They say we know that adaptive arm efficacy increases after the vaccines for SARS-CoV-2. They said, thumbs up, we know we can prove it. We see that in our test as well. The question, curiosity is, will the innate arm stay dumb about this virus, or will it become trained as well? So continuing. So here, what they found was the following. This is the summary of the whole thing. What they found was that once the vaccine is given, Pfizer-BioNTech, first dose, second dose, and I'll talk more about that a little later, they found that the specific and non-specific response. Now, please realize they're talking about innate arm. Innate arm actually does not have a specific response in general. But here they saw that when you have trained the innate arm with the vaccine, or I would suspect even with the correct original infection, the innate arm was trained. It learned to respond to the antigen, which is SARS-CoV-2, and respond with some different modulation of the cytokines. Not only that, innate arm learned to respond differently to some fungi as well, and to the virus as well, and to the other viruses too. So that means those macrophages, for example, who were exposed to the vaccine, they developed some gene expression which made them help against the SARS-CoV-2, but also modulate their behavior against fungi and other viruses too. This is what they call reprogramming. Now, is this odd? Is there something in the vaccine that goes into the nucleus and says, we have a new gene, I'm going to install a new gene over here? No. Anytime when a cell works, for example, let's say we use the example of thyroid gland. When thyroid gland releases thyroid hormone, and that hormone goes and acts on some cell, it would send a message inside the cell, or it would go itself inside the cell, go to the DNA, and open up those genes that are necessary to bring about the response that the thyroid hormone needs. This is a standard behavior of the cellular functions, that what happens is inside a cell, for anything, inside a cell, if you want that cell to do something, then what happens is you send a signal. That signal can either stay outside the cell or go inside the cell. Steroid-like hormones, they go inside the cell. Vitamin D goes inside the cell. On the other hand, there are so many hormones that stay outside. Insulin is a very big example. It would stay outside. It cannot go inside. And it needs a second messenger system inside the cell. Eventually, the message would end up in the nucleus. Most of the time, the things that stay outside, their message is brought into the nucleus via our own proteins. We don't allow foreigners to go into our nucleus and do things. So when the nucleus is told that, hey, cell, I want you to work more, nucleus says, fine, for me to work more, let's say I want to make some more proteins, I'm going to open up a certain gene, part of my DNA. I'm going to open it up. There is a gene there. I'll make messenger RNA from that gene. That messenger RNA will be sent out in the cytoplasm. That would help work with the ribosome, will make proteins, and those proteins would do whatever function is necessary. This is a regular, normal behavior of so my apologies. It seemed like I had my internet froze for a while. Can you hear me? How about now? Can you hear me? Okay. So my apologies. I don't know what happened. So let's continue. So the DNA is nothing but a recipe book. It is a book which has many recipes of making various proteins. And we open the book at a certain page, we copy that recipe in the messenger RNA comes out, and we make that protein, and that's it. That's how the cells function. It's a regular normal function. So back here, they saw that the innate arm, the gene expression in the innate arm as a result of the vaccine was such that the innate arm responded more towards SARS-CoV-2, which we understand it should. And it is fun to understand that innate arm learned to respond to SARS-CoV-2. It's a specific thing, and innate arm is a non-specific arm, and it learned to do that. And it does that for other viruses and infections as well. It's not a gene therapy. Secondly, turned out that innate arm learned to respond, or those genes that got opened up, they helped the innate arm respond better to fungi as well. So remember when we did the children and I want to make sure that I'm still on air and you are able to hear. Okay, so so hopefully you can still hear. So remember when we had a discussion about the children and why do they have lesser of the SARS-CoV-2 symptoms? In there, we talked about, number one, natural killer cells are more. Number two, their respiratory epithelial cells, immune cells, macrophages, and innate arm has better trained immunity. It has learned by attacking other viruses how to work with the viruses. This is the same mechanism here, nothing new. So this is what they found. And so what they saw was those cells that were trained by the vaccine, innate arm, these cells worked lesser against the bacteria. Now, here is a thing. Working less against the bacteria doesn't mean not controlling the bacteria. And this is where they cannot actually tell you that how less means what, because our body can handle these things very easily. And you're seeing people are getting vaccines and they can handle infections. Otherwise, they'll just get sepsis and other things very fast. So one, they saw that the Toll-like receptor 4, when that is stimulated on the innate arm, the response with the cytokines is slightly less. That's what they observed. The observation here is not this, that now you cannot handle a bacteria. The observation is that there is some gene expression in the innate arm that makes innate arm less responsive to gram-negative bacteria and release less cytokines. Now I want to add one more concept here, which is important, and that is here. In general, although debatable, innate memory is considered as a non-specific, short-lived phenomena as opposed to adaptive memory that is long-lived. So this learning that macrophages have done and the NK cells or the dendritic cells have done, it is usually short-lived. We can't expect innate arm to do this for decades, but we hope it knows it. We hope throughout our life, when innate arm handles various pathogens, it continues to learn how to manage them in the future. We do not know exactly how it does it. So here is one observation they had. The second observation was that the innate arm, when we exposed it to viruses with TLR 7 and 8, innate arm's response was also different compared to those who were not vaccinated and who were exposed to the same viruses. Now, is this necessary that this is because of vaccine? Maybe. It is also possible that everybody's response is different type. Question that we should keep answering is, does this innate arm not work anymore? So somebody who reached out and said, well, see, Geert was right. Well, the Geert said that the innate arm would be depleted. There is no concept of depletion of innate arm. There is actually, and I'll show you in the paper, there is actually a good outcome over here that they are saying that innate arm gets trained and responds better. And they're saying that this reduction in response to some things actually helps innate arm become balanced. And I'll have you read that in the paper. It's not me. Become balanced so the cytokine storm does not occur. So continuing, greater response to fungal stimuli. So when they offered fungus to the same cell, which was trained by the vaccine, then those innate arm cells responded with more intensity against the fungi. So that was interesting. It is a modulation that for bacteria and viruses, the response was less intense. To fungi, the response was more intense. Then what they did was, they also checked what kind of response overall is happening. And they saw that after the Pfizer vaccine, this is very interesting. After the Pfizer vaccine, humoral response, now we're talking about adaptive arm, humoral response was little less, but the cytotoxic response, which is actually a good response, was more. So that's actually also a good thing. Then they found innate immune memory and training. So innate arm memory and training is called trained immunity if the innate arm learns to fight better against something, or it is called innate immune tolerance if innate arm learns to not work against some pathogen. This is a 2020 study. There is a study from 2014 in the same direction as well. There are studies since 1940 that was doing this. I discussed this study about a year ago that children respond better because of innate arm, should I say reprogramming, because of continuous exposure to respiratory infections. So how did they do this study? They took 16 people, volunteers, and said, hey, we'll give you vaccine. And after each vaccine dose, after some weeks, we'll take blood from you. We'll look at the cells' responses. We'll look at the antibodies produced, and we'll see what kind of response you're producing. So that's the study. In this, what they did was they took 16 people. They were males and females in there, healthy, mostly health workers, 59 years and below. They took their blood before the doses of vaccine to have a baseline understanding of the blood's characteristics, the antibodies, the type of cells and their activity. Then after the dose 1, they waited for 3 weeks and they took blood samples again, once again to see what kind of cellular response, what kind of cytotoxic response, what kind of humoral response, what kind of innate response is generated by these folks who had these vaccines against viruses, fungi, and bacteria. Then they gave them the second dose and they collected their blood once again 2 weeks later, and they did the same experiment once again on those blood to see what kind of responses were occurring. And they were more interested in seeing the antibodies for RBD, receptor binding domain of the spike protein, and the S protein, that is the spike protein itself. What they found was this. Check this out. They found that after dose 1 of vaccine, IgG was 56 times more compared to the baseline, that is the IgG before the vaccine, which we would expect it to be nothing. It was 56 times more, and after the second dose, it was 1,839 times more. This behavior that they are observing is IgG, which is adaptive arm. So it's not just the innate arm, they actually saw the adaptive arm response as well. And their basic conclusion was, hey, good job. Both adaptive and innate arm learned to work better against the pathogen. Then they also wanted to see IgA, mucus protection after the Pfizer vaccine. And they found 2 kinds of antibodies against the receptor binding domain and against the spike protein. And they saw that after dose 1, the receptor binding domain was 7 times higher antibodies. And after dose 2, the receptor binding domain antibodies were 24 times higher. And against the spike protein in general, the antibodies were 35 times higher after dose 1 and 52 times higher after the dose 2. So what was the result of this understanding? Hey, adaptive arm is working very good. Antibodies are being produced against RBD. They are being produced against the spike protein. All good. Good job. Okay. Then They said, now we were concerned, we were curious, how would innate arm respond? And we wanted to understand interferon gamma. So now we, the cool beans here, level set ourselves about the interferon gamma. I have done this discussion before as well. There is a concept called interleukin 12 interferon gamma axis. I can actually look it up for you right now here. If I write interleukin 12 interferon gamma axis, and you would see a lot of articles about this. This is an old science. It's not new. And what is this science? This science is the following. Here is the innate arm. When innate arm becomes activated, it engages with the naive T cell. We all know that. The naive T cell can now go to the T helper 2 pathway or go T helper 1 pathway. The T helper 1 pathway takes us to the cytotoxic side. And if this pathway is taken, then the T helper 1 cell and the cytotoxic T cell, they produce interferon gamma. Interferon gamma's function is to amplify the innate arm. It happens with every infection if T helper 1 pathway is engaged. This is called interleukin 12 interferon gamma axis. Why? Because the innate arm releases interleukin 12. And in response, the adaptive arm, T helper 1 pathway, releases interferon gamma. This is how they both amplify each other. So how do you think they amplify each other? They amplify each other by opening the genes that are necessary to become active and to release more cytokines and to cause a response. DNA in a cell is not sitting there as a closed book. It is there and actively acting like a brain and functioning. So the DNA gene would open up those genes that need to work to create a response. So this is fine. They actually saw that interferon gamma response was better with the vaccine. Do you know what that means? That means is that cytotoxic side is more active than the humoral side. Humoral side is active as well. They showed that in this diagram here. It is active. But the cytotoxic side is active as well, which gives them the confidence that this creates a balanced immune response. And it helps train the innate arm as well with interferon gamma. Very good. I love it. Then they saw that on the innate arm side, BCG and MMR vaccines, they do gene expressions. So they said, will these new vaccines do that too? And they found that yes, they were training the innate arm as well. This is what is called reprogramming. And this is what they use to say, not only the adaptive arm learns, innate arm learns too. To which then people became nervous and said, hey, we have a gene therapy here. So curiosity on the authors, and I'm picking these things from the paper and I'll go over that paper. Will messenger RNA vaccine also train the innate arm like the live attenuated vaccine types, MMR and BCG? So the answer was yes. It is on page 7 of their document. Then they said that, hey, the tumor necrosis factor alpha, interleukin 1, interleukin 1R, they were reduced from these cells. And they actually talk about it to say this reduction is actually better to quell the cytokine storm. So they say it looks like the immune system learned to respond in a balanced way to this pathogen compared to imbalanced response, which causes people to die. On page 8, they say this is possibly a balancing and protective form from the cytokine storm. And I'll go over those pages in a second. Conclusion on page 10. So now let's look at this as well. I hope you are with me so far. So here we confirm that Pfizer-BioNTech vaccine of healthy individuals induce effective humoral and cellular immunity against SARS-CoV-2. Interestingly, however, The Pfizer vaccine also modulated the production of inflammatory cytokines by innate immune cells. Why is this interesting? Because we know this happens with the attenuated vaccines. We did not know it would happen with Moderna or messenger RNA. These are new vaccines. So these are new things to discover. So beautiful. And they say that the modulation was interesting against virus, fungus, and bacteria. And I went over that. In conclusion, the messenger RNA vaccine induces complex functional reprogramming of innate immune response, which should be considered in the development and use of this new class of vaccines. He said, so when in the future you make more messenger RNA vaccines, just know that it trains innate arm as well. Awesome, that's a good thing. I'm going to skip over all of this and go to page 7. So here is the page 7. On the page 7, look what they're saying. So they're talking about a number of things over here. Intriguingly, the best cellular response after vaccination were against B1351. Can you imagine this? They're saying that the best response of the Pfizer vaccine was against the South African variant. The fact that neutralizing antibody responses against this variant were relatively poor, that may raise the possibility that the protective vaccine effect against this variant may be mainly reliant on cellular side. And we have been talking about the cytotoxic side as the cellular side to be a good side rather than humoral response. Then, look at this. Pfizer vaccine decreased interferon gamma production upon stimulation with TLR7, so viruses. Some viruses, not all viruses. And in contrast, it increased the interferon gamma production. They have it somewhere for bacteria as well, and fungi, for example. Then, besides their effect on specific adaptive immune memory, certain vaccines, such as BCG and measles, mumps, and rubella MMR vaccine, also induce long-term functional reprogramming of cells of the innate immune system. It's an older thing. This biological process is also termed trained immunity when it involves increased responsiveness of the innate immune system, and or innate immune tolerance when it is characterized by decreased cytokine production. And here they're giving a 2014 study. So it's not a new thing. It's not that today we found, oh man, there is a reprogramming happening. No, this is happening far before. Although these effects have been proven mainly for live attenuated vaccines, we sought to investigate whether messenger RNA vaccine might also induce effects on innate immune response against different viral, bacterial, and fungi stimuli. So they were curious that will this new vaccine do that too? And they found out, yes, it would. Surprisingly, the production of monocyte-derived cytokines, these are the cytokines, tended to be lower after stimulation of these cells. And then what do they say here? The induction of tolerance, the reduction in cytokines, towards stimulation with TLR7/8 or TLR4. So one is the vaccines, viruses, the other one is bacteria. Ligands mean binding receptors, by the Pfizer vaccine may indicate a more balanced inflammatory reaction during infection with SARS-CoV-2, and one could speculate whether such effect may be thus useful to regulate the potential overinflammation in COVID-19, one of the main causes of death. Do you see how folks interpreted that and how much nervousness, and how the authors have actually put the words in their paper? On the other hand, inhibition of innate immune response may diminish antiviral response. They say that this TLR7 and TLR8 based response that has less cytokines may diminish the viral response. But here is an important thing to note. There is no baseline to say that, hey, you know what, for the viral response, our immune system, innate arm, should respond with cytokines levels 20. I'm making up a number. And in this case, the level is 10. So we think it is not protective. They do not know that. They just know that there is some reduction in response to some viruses. Does our immune system do that all the time? Yes. We modulate our immune system all the time against various viruses, bacteria, fungi. And we try to respond in a more balanced way. In some people, it does not happen. In some people, they respond in a lower way with tolerance, where they should not be tolerant, and they become victim to the infections. That is immunosuppressed. And in some people, we respond extremely unfairly and with more intensity where we should not. And these folks become autoimmune disorder patients or allergic patients. But normally, immune system continues to balance itself out. So that's fine. It is balancing. These results, now this is the next page. This is page number 9. These results collectively demonstrate that the effect of Pfizer-BioNTech vaccine, go beyond the adaptive immune system and can also modulate innate immune response. Good thing. Then they say, because it modulates the innate response, it is possible that if you give another vaccine, it may interfere with that vaccine's response. And that is a fair comment. This is why this vaccine should be taken separately so that it does not interfere with the other one. Will this reprogramming live there forever? No. It is a short-lived reprogramming, as I showed you here. In general, although debatable, innate memory is considered as a non-specific short-lived phenomena as opposed to adaptive memory that is long-lived. And here, so look at this paper. This paper is 2018. We didn't have the concept of this SARS-CoV-2 pandemic, so it's not somebody sneaking in their paper. This is 2018, and here they're talking about various kind of innate immune responses. It's actually fun to read this paper. In invertebrates, so non-vertebrate people, animals, which like plants do not have adaptive immunity, the protective innate immune response are modulated by previous exposure to infection stimuli, resulting in an increase of a subsequent response to the same or unrelated challenges in terms of increased number of phagocytes, upregulation of gene related to enhanced clearance, and so on. It's a normal behavior in many, many species. And finally, I'm going to go to the page 10, which is their conclusion. So here's their conclusion. In conclusion, our data show that BNT162b2 vaccine, Pfizer-BioNTech, induces effects on both the adaptive and the innate branch of the immunity, and that these effects are different for various SARS-CoV-2 strains. For example, they're better for South African, and they are lesser for 117, which is fine. It depends upon how the immune system got challenged. It also depends upon who is the one. I want to have you read this as well. Check this out. In conclusion, this is a different paper. In conclusion, this is 2018 paper. It is important to remember trained tolerance and potentiation. So he's talking about innate arm. Do not exclusively depend on the priming stimulus. It does not, I can read it for us to say, it does not depend on the vaccine only. Instead, a myriad of environmental factors and other variables affect trained innate memory, including individuals' history of pathogen, antigen exposure, organ and tissue microenvironments, health and metabolic conditions, gender, age, and so on. Intriguingly, the Pfizer-BioNTech vaccine induces reprogramming of the innate immune response as well, and this needs to be taken into account in combination with strong adaptive immune response. This could contribute to a more balanced inflammatory reaction during COVID-19 infection. So when the actual infection occurs, this response that they observed actually allows both arms of the immune system to respond in a balanced way. This could contribute to a more balanced inflammatory reaction during COVID-19 infection, or it may contribute to a diminished innate immune response towards the virus. So they don't know. It could be any of those. Pfizer vaccine is clearly protective against COVID-19, but the duration of protection is not yet known. So this is now just the boilerplate messages. So I hope that this makes sense, and I hope that there was not much of the what is this? The internet issues. Was I continuously reaching you, or did I have more network issues? So if I was able to, I'm going to stop here. We'll do a chit chat in 5 minutes. And if you have any questions related to this topic as well, we'll talk there. But generally, this is actually a good news. They have confirmed some things that are really important for a good, possibly better outcome instead of a negative outcome. This is not a gene therapy. There is nothing in the vaccine to go change the genes. Our cells change the genes anyways to work when they are working. Then they close down those genes, or those cells die and the new cells are produced, and so on. So it's nothing weird. This is normal things. Since 1940, we have been looking at those things. So with this, thank you very much. Do me a favor. Please like, subscribe, and share. And if you would like to support this work, there are 3 links in the description. One is for buying me a coffee if you don't want to use PayPal, or you can use PayPal. That link is there as well. Or you can become a patron to continue to support this work for others in the future as well. And with this, I want to thank Margaret for once again a healthy support, healthy donation yesterday. So Margaret, thank you very much. You have been consistently so helpful for last so many months. So thank you everyone else as well who has supported, and I would see you in a few minutes.